Hello everyone. This week we're going to talk about microbes of the urinary tract. Traditionally, the urine was considered a sterile body fluid, but now we know that voided urine is not sterile and it is going to have normal, flora within it. If you think about it, urine is a filtrate of the blood, and our blood is sterile, so that's why, for the longest time we thought that urine is sterile. But they recently found that the urinary bladder itself does have microbes within it. And if you think about it, when the urine gets voided and it passes through the urethra, they also discovered that the urethra itself, the walls of the urethra, does have also microbes. So as someone voids their urine, these microbes will go ahead and get released along the way, so that urine is not going to be, that voided urine is not sterile anymore. Therefore, we now know, right, that it's absolutely normal to have an amount of bacteria in the urine. So how can we tell whether someone has a urinary tract infection or not? We're going to talk about the numbers. So they look for, there's a range, and so as long as you are within the range, like there are, yes, some bacteria within your urine, but you're still within that, that range, then you don't have a UTI. But if they found out that, you have a whole lot of bacteria and now you've surpassed that normal range, now this is an indication of a UTI. And of course, they also take a sample of, the urine, and they go ahead and they culture it in the lab, and they look for certain, bacteria that we know is common, right, in causing UTIs. So they go ahead and they also look for that. And so therefore, when we find bacteria in someone's urine, like we could assume there are pathogens within that batch of urine that was just collected. And so there are two types of analysis that we do on someone's urine sample. There's quantitative analysis, and this is where we go ahead and we try to figure out the number of bacteria per mil. And how do we do that? Well, we go ahead and we culture the urine on a plate, and we go ahead and we start counting the colonies. And so we're going to go over that in detail. And then there's also qualitative analysis. We go ahead and we look at the features of the bacteria that's growing in the urine. Because as I said, there are certain bacteria that we know are the ones that usually cause urinary tract infections, such as E. coli. So E. coli is like the #1 bacteria responsible for urinary tract infections. So we go ahead and we do some biochemical tests. We do, some other tests that we could, figure out whether there is, let's say, E. coli or not. I mean, as I said, E. coli is the most common, but are there other bacteria that also cause UTIs? Yes. We can go ahead and we test and we take a look at the features of these bacteria. Let me show you guys a way of how to do quantitative analysis of the urine. So let's say you just collected some urine sample from your patient, okay? This urine sample, we must go ahead and dilute it first before we go ahead and we streak that urine on, some nutrient agar plates that we have. We must dilute it first. So let's say that you go ahead and take 1 ml of that urine sample, and you add it to 99 mils of sterile saline. Of course, after you add it, you have to shake it and mix it first before you start plating it. But let's just think of, okay, what does that mean? 1 in 99, that means that I did a 1 to 100 dilution. Now, if I go ahead and plate 1 mil, okay, if I go ahead and plate 1 mil of that dilution that I just did, what does that mean? It means this plate is a 10 to the -2, or you can think of it as 10 to the 2, right? Dilution. And if you go ahead and plate 0.1, what is 0.1? 0.1 is another 1 over 10. So that means that kind of count your zeros, 1, 2, 3, that means that this plate right here, its dilution factor is going to be 10 to the negative 3, or you can just think of it as 10 to the 3. Then what do we do? So after you go ahead and you streak your plates, well, you're going to go ahead and start looking right for, for growth. So let's say right here, you're like, these dots that I'm just kind of showing you are representing like, the colonies. Well, you're going to go ahead and start like, counting these colonies. So let's say you counted like 70, okay, 70 colonies. So that means that, okay, how much bacteria do you have here? So that's 70 times 10 to the 3, but go ahead and put it in scientific notation. So that's basically like 7 times 10 to the 4 bacteria per mil. Now, is this considered like, a UTI or not? We're going to talk about it. Is this considered like a high amount of bacteria and we can say that the patient has a UTI? I'm going to talk to you guys about, that number in just a few. Now, there's another way actually of culturing a urine sample, and this way is so much easier. Now, instead of diluting the urine sample of your patient, kind of like how we did previously, take like 1 ml, dilute it in 99 ml, which is going to take, time. This is so much easier. You go ahead and you grab a calibrated loop. So this right here, you guys, is we refer to it as a calibrated loop. And this calibrated loop right here, it's actually calibrated to measure 1 microliter of any fluid sample. So if you take this calibrated loop, let's say I have here, my urine sample, okay? So here is my urine sample, and you go ahead and you basically dip that calibrated loop, okay? You dip that calibrated loop in here. How much are you going to be collecting? How much urine are you going to be collecting? You're going to be collecting 1 microliter. So this calibrated loop will measure exactly 1 microliter into 1 microliter of the fluid. So if you dip it into the urine sample, it's going to collect 1 microliter of the urine. Now, what is 1 microliter in terms of milliliter? Well, you're going to divide by 1,000. So let's say this is like 1.0. Let me just write it a bit better. So here we go. So as I said, you guys, it's going to measure 1.0 or like 1, right? Microliter of urine. You're going to divide by 1,000. So you're basically going to move it. You're going to move your decimal towards the left. Okay, 3 spots. So that means, that it's going to collect 0.001 mils of urine. Okay, so 0.001 is the same thing as saying 1 microliter. Okay, so that means right there that you've diluted your sample. Let me first show you after you dip it into the urine sample. Okay, what do we go ahead and do? So you dip that calibrated loop. As I said, it has to be a calibrated loop, not just any loop out there. No, it has to be one of these calibrated loops. And you can see like right here, their loop is super like small compared to any other loop that we use. So you go ahead and you grab this specific calibrated loop and you dip it into the urine sample. And then you go to your nutrient agar plate and you streak, 1 line down the middle of the plate. Okay, and then what do we do? With the same loop, you don't go redip, you don't do anything, right? You don't sterilize or anything. After you've streaked 1 loop down the middle, you go ahead and you take that same loop and you do zigzag, okay? and you have to, like, your zigzag needs to be like pretty close. Okay, so you go ahead and you zigzag. You don't go back and forth, so you just like zigzag all the way down the plate and you're done. So you do these two streaks. Now, of course, this loop has to be sterile to start off with, right? So you're using a sterile calibrated loop. You dip it into the urine. It's going to collect 1 microliter or 0.001 mils of that urine. You streak once down the plate, and then using that same loop, you go ahead and you do that zigzag pattern. Okay, and then you go ahead and you incubate your plate, of course. Now, is there another way to write 0.001 mils? Yes, 1 times 10 to the negative 3 mils, right? So 1 times 10 to the negative 3 is the same thing as saying, 0.001 mils. It's the same thing as saying 1 microliter. So if you guys notice, okay, what did we just do? What we just did is we went ahead and we diluted our urine sample. You've diluted it by 10 to the 3. Okay, and so do you see how this is so much easier compared to taking a urine sample, adding it to saline solution and diluting it this way? This was just one step. All you did was dip that calibrated loop into the urine and you went ahead and you plated it right here. Now, this plate right here is now a 10 to, it's a 10 to the negative 3 plate or a 10 to the 3 plate. So all what you're going to do is after you've incubated it, okay, you're going to go ahead and you're going to see the growth. And one more time, you're going to start counting. You're going to start I'm just putting here some colonies on the plate. So you're going to go ahead and you're going to start counting these colonies. So let's say you counted 72 colonies. So that means you have 72 times 10, to the 3 bacteria per mil. And again, go ahead and write it down in scientific notation. So that means 7.2 times 10 to the 4 bacteria per mil. So do you see how we went ahead? We actually, diluted our urine sample all we did was use that calibrated loop. So it's so much easier, right, to use that calibrated loop, to culture someone's urine. And then after you go ahead and you streak one down and then the zigzag, you incubate the plate and then you start counting the colonies. And this plate, as I said, it's going to be a 10 to the 3. And you're going to figure out how much bacteria does someone have. Now, again, is this number considered a UTI or not? We'll go ahead and discuss that in a few. Now, for the qualitative analysis of the urine, what we usually do is we take the urine sample, we go ahead and we transfer. So let's say here you have a batch, okay, of urine. We go ahead and from that urine sample, we take about 10 mils of it and we put it in a centrifuge tube. That centrifuge tube will end up being placed in a centrifuge machine, and the centrifuge machine is going to spin the urine super fast, and it's going to go ahead and it's going to separate, okay? Separate the urine. So it's going to separate. When you go ahead and do that, you're going to end up with a pellet. Okay, so like the bacteria in the urine is actually going to go ahead and kind of like precipitate at the bottom, and then you're going to have like the fluid. Okay, you're going to have the fluid up at the top. Okay. We don't want the fluid part. What we are really interested in is really that bacteria pellet that you're going to get at the bottom of the tube. So you go ahead and, as I said, after you've centrifuged it, you're going to have that separation right here. you're going to pour the fluid out, okay? So you're going to throw out like most of it, or basically like pour out most of it. You're going to decant the tube, and you're going to take that pellet that you're going to get, and you go ahead and you like break it up, and you're going to take a sample, okay, of that pellet, and you're going to streak it on an EMB plate. You're going to streak it on a blood agar plate, and you're also going to dip it into the thioglycolate broth. Now, we know that EMBs are both selective and differential plates, so they're going to only allow gram-negative bacteria to grow on it, and they're selective for lactose fermentation. If you see like pink color or if you see a green color, that would mean that the bacteria can ferment lactose. Blood agar plate is like an enriched type of media, so it's not selective, but it's going to, it's an enriched, so it's going to allow all sorts of like different bacteria to grow on it. Blood agar plate is not so good though for fastidious bacteria, but this is kind of why we also use like a blood agar plate, because it's an enriched type of media. Now it is differential. It's going to tell you whether the bacteria can go ahead and hemolyze blood cells or not. And then here, the reason why we're also dipping it into a thioglycolate broth, because if you recall, we talked about thioglycolate broth before, and we said that you can figure out whether your bacteria is anaerobe, aerobe, facultative, etc., based on where you see the growth, right? So like if the growth is all the way up at the top, then you know that your bacteria is an obligate aerobe. If it's like throughout, then you know that your bacteria could be a facultative anaerobe, for example. So the thioglycolate broth is going to definitely help you figure out the oxygen requirement of the bacteria. And this is going to help you identify, right? Like these tests are going to help you identify and figure out characteristics about that bacteria that this individual has in their urine. So that's why it's a qualitative test. You're going to figure out some characteristics, right, about the bacteria found in this patient's urine sample. Now, some extra notes, right, to know about the microbes of our urinary tract. Women are more likely to get a UTI compared to men, so they're more likely to get a UTI compared to men, and there are anatomical reasons for that. One is the women's urethra is much shorter in women compared to men. So the urethra, it's the tube that's going to eventually like the urine gets released out of the body through the urethra. So the women's urethra is much smaller, much shorter in size compared to men. So therefore, they are more susceptible to getting a UTI. Number two, the anal opening also is closer to the urethra in women compared to men. So if a woman goes ahead and kind of like wipes from back to front, if you think about it, the anal, this is where the fecal matter comes out, and feces does have bacteria, including E. coli. So, if this happens and the E. coli gets, let's say, as the woman is wiping, wipes from back to front, some of that E. coli, right, from the fecal matter can make it, to the urethra opening, and therefore, as I was saying, E. coli is the most common, bacteria to cause UTIs. And because the anal opening is closer to the urethra in women compared to males, again, women are more susceptible to getting UTIs. Now we talk about the quantitative analysis, right? So we said how to go ahead. We talked about two ways, right? We talked about how we can dilute the urine via two ways. You can just, take a sample, take like 1 ml of the urine, dilute it into, 99 mils of saline, and this is how you go ahead and dilute it, and then you streak the plates. Or we said the easier way is to use a calibrated loop, just dip it into the plate and then dip it into the urine and then go ahead and streak it. And we said that after you incubate your plates, you start counting the number of colonies. Now, how much, right? How many really bacteria per mil are we looking for to consider, that this individual has a UTI? We're looking for something like more than 100,000, okay, bacteria per mil. That would tell us that this individual does have UTI. Because as I said, you could find bacteria in someone's urine, but how much is like, okay, too much? And therefore we consider it a UTI. Anything like more than 100,000 bacteria per mil will be considered a UTI. And if we want to write it, in scientific notation, well, it's going to be 1 times 10 to the 5, right? So here 1, 2, 3, 4, 5 zeros, right? 100,000. So you can write it as 1 times 10 to the 5 bacteria per mil, okay? If you want to write it in scientific notation. So therefore, if we find 1 times 10 to the 5 bacteria or more, right, than 1 times 10 to the 5 bacteria per mil in someone's urine, we are going to consider it as, this individual does have UTI. Now, let's say someone has like 10,000, anywhere between like 10,000 and 100,000, which is pretty, like a lot, right? And they're like right there, let's say like, at the 100,000, limit. This could actually be a false positive. And the patient is still negative for a UTI. So the patient is still negative for UTI even though they have like that much bacteria in their urine. I mean, it's not really, an individual shouldn't really have like that much, okay, bacteria in their urine. So what could be some reasons why someone has such a high number of bacteria even though they don't have a UTI? Well, maybe the patient didn't wipe properly before giving the urine sample. If you've ever, given a urine sample, you kind of know that they give you a sterile cup, by the way, so they must give you a sterile cup because they want to make sure that this cup doesn't have bacteria in the first place. Otherwise, your sample will be contaminated, so they must give you a sterile cup. Number 2 is they give you like a wipe, right? Like it's like, a disinfectant wipe, and they ask the patient to go ahead and wipe before giving the sample. And the reason why they ask for the patient to wipe themselves before giving the sample, because there is bacteria on the skin, right? So they're trying to eliminate, right? They're trying to eliminate and reduce the number of the bacteria on your skin so it doesn't, give you a false positive. So if someone forgot to wipe properly or didn't even wipe, that could be a reason why their sample might have a whole lot of bacteria in it. Number 2, they also ask you to give them a midstream urine sample, a midstream catch. What does that mean? So when they give you that cup, the individual is not supposed to urinate right away into the cup. They're supposed to actually like urinate a bit, right? And allow some of that urine to get voided first, like in the toilet, and then go ahead and catch, some of that urine sample. Like, so that's why we call it a midstream catch. And the reason why they don't want you to urinate right away into the cup, because that first, those first few mils, okay, of urine coming out, it's going to collect like a lot of the bacteria in the urethra, right? So that's why you want to actually kind of like let some of that urine first like get voided, okay, where you are going to have like most of the bacteria from your urethra coming out, and then you want to go ahead and kind of catch it midstream. So you've made sure, that most of that bacteria was voided right into the toilet, from your urethra. And then, number 3, like, what could be a reason why you could have like a whole lot of bacteria in your sample? Maybe the sample was not tested in a timely manner. So the lab kind of kept your, your urine sample for a pretty long time before they go ahead and they test it. And this could lead to whatever bacteria is in your urine to go ahead and multiply, multiply in your urine, and therefore you can get like a huge sample of bacteria in your urine, even though you don't have a UTI. But as I said, like this is very rare, right? They're usually pretty good in not making mistakes like this. But if they do and they realize, oops, like the batch has something went wrong and they didn't test it in a timely manner, they're going to call you up and ask you to come back and give them a new sample. Okay, so again, like if something goes wrong, like with any of these three like steps that we've talked about, the amount of bacteria in your urine sample will be high, even though you do not have a UTI. So you have to make sure, especially with those two steps, that you do them properly when you're giving a urine sample. Okay, we said that we can go ahead and use the calibrated loop plate so that we can go ahead and dilute someone's urine. And we said that we use a calibrated loop that's, calibrated to hold exactly 1 microliter of the urine sample. Now, again, what is 1 microliter in terms of mil? Well, it's 0.001, right? Which is the same thing as saying what? Just the same thing as saying 1 times just the same thing as saying 1 times 10 to the negative 3, right? So you went ahead and you diluted someone's urine by 10 to the 3. So the dilution, right? What type of dilution? We're basically doing a 1 times 10 to the negative 3 dilution. And so if you think about it, if we are using a calibrated loop plate method, how many colonies would we want to consider that this individual has a UTI? Well, to have like 100,000, which is, as I said, like, if you find like more than 100,000 colonies, 100,000 bacteria per mil in someone's urine sample, that would be considered a UTI. So you would want at least to find what, like 100 colonies. Why? Because if you think about it, if you see 100 colonies, right? So let's say here is your 10 to the negative, here's your 10 to the negative 3 plate, okay? And you found 100 colonies in here. What does that mean? It means that you have what? 100 times 10 to the 3 bacteria per mil. Okay, what is that in terms of, what is that in terms of like scientific notation? Well, it's the same thing as saying what? 1 times 10 to the 5 bacteria per mil. Okay, what is 1 times 10 to the 5 bacteria per mil? That's the same thing as what? Saying 100,000 bacteria per mil. And we just said that if someone has like, more than that, like more than 1 times 10 to the 5 bacteria per mil, we're going to consider that to be a UTI. Okay. Now for the qualitative analysis, we do use the EMB plate. As I said, gram negatives will grow on it, only gram negatives. It's differential for the lactose fermentation. So you get that pink or green color. It will tell you that the bacteria can ferment lactose. We also use the blood agar plate. It's an enriched type of media, so it's not selective against it's not preventing any bacteria from growing on it, but it is differential for hemolysis. You can figure out whether it's an alpha, beta, or gamma type of hemolysis. No gamma, no hemolysis, alpha, partial, beta, complete hemolysis. We've extensively talked about the blood agar plate results before. And then we also use that reducing media, that thioglycolate broth, which is reducing media for like oxygen. It does kind of like, we add the chemical to it to help like remove that oxygen from the tube. There is still going to be some oxygen in the thioglycolate broth, but not a whole lot, especially towards the bottom of the tube. And that's why, depending on the growth pattern within the thioglycolate broth, we can figure out whether the bacteria, is an aerobe versus an anaerobe, etc. And as I said, E. coli is the most common, right, bacteria that causes urinary tract infections. Do we know some features about E. coli? Yes, we do. We know that it's a gram-negative. We know that it's a bacillus in shape. We know that it does ferment lactose. We know that it's a facultative anaerobe, so it should grow throughout the, the thioglycolate broth. So that's why we do these tests, right? Because we want to know whether the individual has an E. coli, causing their causing their UTI infection or not, or if it's some other type of pathogenic bacteria. So for this lab, you guys are going to take a look at some results and figure things out. So right here, you're given like those two dilution, plates. Like this is a 10 to the 2 dilution factor plate. This is a 10 to the 3. Go ahead and kind of take a look at the number of colonies and figure out, figure out the number of bacteria per mil this individual has, and figure out whether this is considered a UTI or not. And then right here, again, let's say this is the calibrated loop plate, and you guys see how we usually just like streak once, and then we go ahead and we do like the zigzag pattern. And again, this is considered what, like a 10 to the, remember, a 10 to the 3 plate. So if you have 95 colonies, again, figure out how much would that be, bacteria per mil. And then here, let's say you've inoculated the urine sample on an EMB plate, on a blood agar plate, on on a thioglycolate broth, and then go ahead and figure out the characteristics, okay, of that bacteria that you've just collected.